much. And well, thank you for the introduction, and also thanks Beppe, of course, for the invitation. I'm very happy to be here today, and from the title you probably already got an idea that we'll change gears a little bit now. So I know that you've heard excellent talks about messenger RNA vaccines, and what I will be talking about today is my first scientific love, which is short interfering RNA. So short interfering RNA is a little bit different from what we usually do with messenger RNA. This is also my disclosure statement that I have to show. And with short interfering RNA, we usually, the idea is that we try to downregulate gene expression, whereas with messenger RNA, we obviously try to induce gene expression. And so one of the reasons why we're interested in short interfering RNA is because there are many diseases where a therapeutic downregulation of an upregulated gene or an oncogene or other genes that are pathologically expressed can lead to a therapeutic effect. And many of these diseases are in the lungs. So there are many lung diseases that currently don't really have any good therapeutics, but where we just try and palliate the symptoms. And so the idea with RNA therapeutics is that we can potentially really look into the underlying cause and really come up with a therapy that is more upstream of palliating symptoms. And you probably know that there have been several approved nanomedicines and also RNA-based medicines. Not all of them are nanomedicines. And depending on what administration route we choose, most of these RNAs still end up in the liver. So you probably also know that there are RNA therapeutics that are directly injected into the eye or intrathecally, for example. And so this is a similar approach of what we've chosen, of using a topical, using a local administration route for inhalation, because we know that the lungs are directly accessible via inhalation. But the problem with the currently approved formulations for RNA delivery, the currently approved nano formulations, is that they don't withstand the shear stress during nebulization. So you see that during nebulization, or depending on how we formulate them, they tend to rupture, they tend to be disrupted by shear force or temperature stress. And this is why we are looking into new formulations for RNA that can really be delivered to the lungs via inhalation. And while we do some LNP work, we also focus very much on new biomaterials that are polymer-based and amphiphilic polymer-based. And you may know, particularly if you are in polymer science, you know that we usually in polymer science design large libraries and synthesize large libraries of materials, and then maybe we come up later with one single candidate that actually works. So this work is often done by trial and error. And our approach is now to use more simulation-based work and machine learning to use a lot of the knowledge that's out there already, and then define structures that will be useful by a more sustainable approach and by a more rational and targeted approach of synthesis. So why are we interested in pulmonary delivery and inhalation? You see that, as I mentioned before, the lung is an organ that can be reached directly by inhalation. So we can use a local administration route, we can use a non-invasive administration route, and we can circumvent the first pass effect in the liver. And with that also decrease systemic side effects. If we want to treat the lungs, it doesn't make sense to deliver a lot to the entire body, but it makes a lot more sense to directly deliver to the lungs. And with this local administration, we can increase the retention of the drugs and also the retention time. And we can make use of the physiology, the special physiology of the lungs, both for a route of entry, so both for in order to deliver through the lungs, if we just want to use the lungs as a port of entry. But in our case, for most of our approaches and the diseases that we target, they are really happening within the lungs, so we usually deliver to the lungs and not through the lungs. And so in that case, we can make use of the large surface area. So the surface area that we have in the lungs is about 100 square meters. That's about a 2-bedroom apartment, maybe not in Barcelona, also not in Munich, but it's a large area that we can target. And we also don't have serum proteins within the lung, the air side of the lungs, which is a big advantage for RNA-based therapeutics. But of course, we also have barriers that need to be overcome within the lungs. In the upper airways, we have mucus, and nanoparticles tend to get stuck in the mucus, and in the lower airways, we have surfactant, and surfactant is nothing other than phospholipids, and this, to some extent, already explains why LNP-based materials sometimes show some disintegration and stability issues within surfactant, because you see now a competition between the different phospholipids. So, as I mentioned before, short interfering RNA has a bit of a different mode of action. So you see that, of course, we still need to deliver the RNA inside the cells. RNA is a large, negatively charged macromolecule, so it cannot enter cells by diffusion. It's simply too large, and the diffusion coefficient is way too small, and it's negatively charged like the surface area. So we need to either exploit endocytosis via receptor-mediated uptake, or we need to encapsulate into a nanoparticle that then can be endocytosed by adsorptive endocytosis, or of course also using targeting ligands for receptor-triggered uptake. But in any case, these particles, once they're inside the cell, they need to be, they're taken up into these endosomes by endocytosis, and they need to escape the endosome. So endosomal escape still is a very big hurdle in RNA for all RNA therapeutics, and also for the approved ones. And then the RNA, in our case, needs to be incorporated, what's called the so-called RNA-induced silencing complex, abbreviated as RISC. And in this RISC, with the short interfering RNA, binds to base complementary messenger RNA, and now it causes endonuclease activity within RISC to dice and to chop up the messenger RNA. And so with that, we lose the message, and if the message is lost, then obviously it cannot be, a protein cannot no longer be expressed or formed alongside the messenger RNA. So we see a downregulation in gene expression of our targeted gene. But of course, it's not as easy as you see in this cartoon, but we have different challenges, as I mentioned, for cellular uptake and then for endosomal escape. And we've looked into different lung diseases where RNA delivery could be used therapeutically. And one of them is asthma, allergic asthma, which really has a large population of patients, and 5 to 10% of them cannot be treated well with currently available therapeutics. And therefore, we thought maybe this is something where we can not just palliate symptoms, and this is exactly what the current therapeutics do, but maybe we can really go one step above and see what's really the driving factor that we need to downregulate to have a better therapeutic effect. And this slide is very busy in immunology, so depending on what your background is, you may or may not appreciate this. The big story is that you see in green this activated T cell. And T cells play a big and important role in asthma. It's an eosinophilic disease, but this eosinophilia is actually driven by Th2 cells. So what we usually try to do for treating asthma is that we try to deliver RNA into T cells. And if you've ever tried to do that, it's quite tricky. So T cells don't express caveolin, for example. So you heard in Dan's talk earlier that caveolin drives or mediates endocytosis, but T cells don't express caveolin. So we already have one entry port blocked, essentially, not available. But you know from HIV infection that uptake via receptors in T cells works quite efficiently. So we figured what is a receptor that we can target in T cells that will trigger the same uptake. And we found in very old literature that upregulated or activated T cells upregulate CD71, which is nothing other than the transferrin receptor. And transferrin receptor has been used largely in tumor targeting. So we figured that most probably we should be able to also exploit this for RNA delivery into activated T cells. And this is exactly what happens in allergies. So you see that an allergen is presented by an antigen-presenting cell that then causes a naive Th0 cell to differentiate into a Th2 cell in green. And then the transcription factor GATA3 in Th2 cells is upregulated to secrete all the Th2 cytokines. And all of these IL-4, IL-5, IL-13 then cause this entire orchestra of inflammation, and then cause downstream effects that then cause all the symptoms of asthma, such as shortness of breath and mucus overproduction, bronchoconstriction. And so this was not exactly our idea, but many other people had also looked into downregulating single cytokines for a therapeutic effect. But they saw that if you downregulate one of these cytokines, the other ones will be upregulated. So this is not exactly the solution yet. So we and others thought that if you look at this orchestra of inflammation, and you can knock out the conductor of this orchestra, so knock out GATA3, then you should be able to see a silencing of the entire orchestra. And this is exactly what happened. So our idea was to deliver short interfering RNA against GATA3 into these activated T cells, and with that really see a downregulation of all of the pro-inflammatory cytokines and processes in asthma. And so for proof of concept, we initially started working with polyethylenamine. I'm sure many of you used it for proof of concept work. It obviously is not an innovative polymer. It's non-biodegradable. It causes toxicity. We used a low molecular weight 5 kilodalton one version in order to keep it as well tolerated as possible. Attached transferrin to it with a bifunctional linker, and then applied it, or initially did some ex vivo work in primary T cells that looked very promising, to then move into in vivo studies where we used a typical asthma protocol of using ovalbumin as a model allergen, where then we exposed these animals to inhaled ovalbumin after we sensitized them by intraperitoneal injections. And you can see them in our old exposure tower, where the animals sit there, and considering that mice are obligate nose breathers, if they're sitting in these exposure tubes, they will inhale the ovalbumin solution and develop all the symptoms of asthma. We just bought the more modern and more instrumentized version of the tower, and are very happy to now move on with this. But right around the time that we had the best initial results showing that if we use these transferrin-targeted PEI and we use fluorescently labeled siRNA, we can really see a significant uptake in T cells in the inflamed animals. And we don't really see a difference, any difference at all for the healthy animals that simply inhale sodium chloride. And we showed that this was really a specific uptake in T cells in the bronchoalveolar lavage fluid. This is right around the time that I moved back to Germany and was struggling with getting animal protocols approved. So essentially, due to the lack of approved animal protocols, we decided to start working with what's called precision-cut lung slices. And you already heard about this technology on the first day, and Joschka Müller presented very nice work in precision-cut lung slices yesterday as well. The technology works in a way that we have ethics approval to use tissue from the hospital that is then filled with agarose and sliced into very thin slices. And we can then punch distinct areas of tissue out of this lung tissue. And the beauty is that in this lung tissue, we still have all the cell types available. So it's better than any co-culture model that you could think of, and in my opinion, also better than organoids, because you really have real human tissue. And in this real human tissue, there are still a lot of T cells. And then we can use the same protocol that we use for ex vivo and PBMCs, where we use dynabeats against CD3 and CD28 to activate the T cells, and then really see an asthmatic phenotype and see an inflammatory Th2 phenotype in these human lung tissues. And then, of course, we can do all of the transfection experiments and all of the treatment experiments that we're interested in, and all of the readouts you could possibly think of. You have the tissue, you have the supernatant, where you can look into cytokines and tolerability of the treatment, and then, of course, also downstream effects of GATA3 knockdown. And so in these experiments, we showed that with our transferrin-targeted approach, we see a very strong downregulation of GATA3 in the tissue, and also a downregulation of these Th2 downstream cytokines. So we're now looking into further moving this into a more animal experiment, but we're also very interested in really clinically relevant dosage forms. So being a pharmaceutical technologist by training, I'm very interested in really making dosage forms that can be applied by patients. And we're interested in dry powder inhalers. So dry powder inhalers have the advantage that they can be stored at room temperature. So with your RNA storage, I think during the pandemic we all learned that this is a big issue, big concern. And the advantage of a dry powder inhaler is that you can store it at room temperature, and you can, by spray drying, you can form this nanoparticle suspension into a solid dosage form that physically, chemically, and also microbiologically is a lot more stable than any suspension could possibly be. So we use this nanoparticle suspension. We dilute it in an excipient solution, so we usually use trehalose, mannitol, or lactose, and then we spray dry this into what we call nano and microparticles. So I mentioned before that we need nanoparticles for adsorptive endocytosis, but if you want to deliver particles to the lungs, you actually need microparticles. So you need an aerodynamic diameter of about 1 to 4 micrometers, and with a nanoparticle, you wouldn't really reach the deposition within the lungs that you would need. So what you see in this SEM image is microparticles that are made out of many nanoparticles glued together by this amorphous or crystalline matrix, sugar or sugar-alcohol matrix. And with that, you can really achieve the deposition within the lungs that you need for a therapeutic effect. And so then, of course, I'm sure you ask, if you spray dry, are these nanoparticles still bioactive? And yes, they are. So we're able to show that for the transferrin-targeted particles, particularly the mannitol formulations, which are crystalline, work very well. For some other formulations, amorphous matrices such as lactose or trehalose work a bit better. And right around that time, the pandemic hit, so I think everyone in pulmonary research, of course, was thinking we have to try and do something here. So we had already started talking with a virologist in the summer of 2019 who was actually interested in a respiratory syncytial virus. But in January 2020, we, of course, changed gears and moved everything into a SARS-2 research. And the beauty of our precision-cut lung slice model is that we use human tissue. So we have started to do animal experiments in the ACE2 mice, and we saw that the mice were essentially dying from encephalitis rather than from their pulmonary infection. And we figured that at the time, the animal models were not great. We didn't have access to the hamster model, unfortunately. But we figured it should be more or less easy to infect human tissue with SARS-2, and it was. So we used the precision-cut lung slices that we obtained from the hospital and optimized an infection model with a wild-type virus, and were able to get very clear replication of the virus, and then screened different siRNA sequences and their modifications in collaboration with our virology collaborators in Munich, who then looked into all of these different sequences, different targets within the viral genome, and in the end were able to identify an siRNA sequence and modification 03 asterisk that was able to downregulate viral gene expression in the human tissue by almost 90%. So you see that also our virology collaborators initially proposed a prophylactic setup. So we pre-transfected the tissue, then infected, and checked if we could avoid for this infection to happen. That worked very nicely. So now, of course, it's our task to see if we can rescue infected tissue, and then, of course, rescue infected animals later on. But I mentioned earlier that we're not really interested in PEI. We're not really interested in trying to bring PEI to the clinics, but we're interested in identifying new materials. And so many years ago, we started looking into oligospermines. And these oligospermines have the advantage that they're made out of an endogenous polyamine, and that they're biodegradable. But the problem with these oligospermines, with the bispermines and tetraspermines, was that they were not very efficient in encapsulating RNA. Particularly short interfering RNA, you have to consider as a very rigid double-stranded RNA that cannot be very easily encapsulated into a polyplex by electrostatic interaction. So one of my previous students had the task of making polyspermines. And I asked him to synthesize these materials based on polybeta amino ester backbones, which is a very simple Michael addition reaction. And he started doing these experiments where he used triboxspermine to attach it to the backbone, to the polybeta amino ester backbone, and essentially received different molecular weights of this polyspermine material. And he started working with the polybeta amino ester polyspermines, and we saw very good uptake into the cells, but no gene silencing. So we figured it must be about endosomal escape, and he had an idea that he said, why not use the Boc-protected version? So before we deprotect them, they are more amphiphilic, or they're slightly more hydrophobic. So why not use this and then exploit the fact that the acidic environment in the endosome to see if this will help endosomal release. So he compared the Boc-protected and deprotected versions for RNA encapsulation, and you see that with the Boc-protected version, considering that there are no primary amines, the encapsulation shown in green was not as good as with the deprotected version. And then, of course, we also saw very negative zeta potentials until higher excesses of polymer over RNA. But he still went on with the experiment, and he showed that while he didn't see any gene silencing in our GFP expressing lung cancer cells, he was able to see this gene silencing with the Boc-protected version. So his idea certainly was good. He confirmed this by using chloroquine to really force endosomal escape, and that helped in both cases. But he was quite convinced that with the Boc-protected version, he seemed to see a better endosomal escape. But so then he moved on to a more therapeutic model rather than just GFP silencing and downregulated mutated KRAS in lung cancer cells, and was able to show that it also causes, it has an effect on cell invasion and motility. But my idea was a bit different, so I said, how about using two different monomers for the synthesis? Rather than using this Boc-protected version, you use the deprotected spermine, but you also include a monomer with a hydrophobic side chain. And so I suggested to use oleic acid, or in this case, oleoylamine. And so with these two different monomers, we can tailor the ratio. So we can make different polymers with, for example, 10% oleoylamine, 90% spermine, which are mainly cationic, or we can make a 50-50 polymer, for example, that then is a lot more hydrophobic and really amphiphilic. And he did all of the characterization, showed that in terms of sizes and zeta potential, it didn't really have much of an impact. And in terms of cellular uptake, it really mattered. So it seemed like the most cationic polymers shown in pink showed a very good uptake. And then, of course, also these dark red bars show very good uptake for the 50-50 polymer. But if you look into gene silencing on the lower panel, you see that even though the cationic polymers were taken up very efficiently, they apparently didn't make it out of the endosome because there was no gene silencing in comparison to the negative control siRNA. But the blue polymers with the 30-70 ratio that were more hydrophobic weren't taken up very efficiently into the cells, but then had a very good gene silencing efficacy. So the little amount that was taken up made it out of the endosome, apparently. And now with the 50-50 polymer, he was able to show 95% gene silencing, which was comparable to a commercially available transfection reagent. But this, again, is just in a 2D cell culture in a GFP-expressing model. And in the lungs, we deal with different barriers, as I mentioned before. So we used air-liquid interface cell culture. using kalu cells that secrete and produce a lot of mucus and make a very strong barrier against nanoparticle diffusion. So we grew these cells until the point that they form tight junctions, produce mucus, and then added the particles to the so-called air side. And interestingly, we still saw very good gene silencing efficacy, not the 95% we had seen before, but a good 60% gene silencing also in this more sophisticated and more relevant model. And now I told you earlier that rather than screening all these different polymers, we're more interested in better understanding how the polymers interact with the RNA so that we can really design and synthesize the polymers that interact well, encapsulate well, release at the right time, and can also diffuse through mucus. So if you compare these molecular dynamic simulations of the different polymers that we've synthesized, you see that at a low, so at a high cationic content, there are a lot of polymers that don't really participate in the interaction with the RNA. If we increase the hydrophobic amount, you still see some of these what we call non-productive micelles, because you see these 3 ones in the upper left that are not participating, they're not interacting with the RNA, shown in green and blue. But if you increase this further to 80% oleolamine and only 20% spermine, you see really good encapsulation and very good participation of all of the polymer into a more defined formulation. So we're hoping that with these simulations in the future, we'll be able to use the simulation work as well as the experimental bench work to feed our machine learning algorithms. And some of the work we've done in the meantime has already revealed some new materials that have outperformed all of the other ones that we synthesized by trial and error in the past. Now, I just want to give you a very short comparison to a different type of polyspermines. And so these were polyspermines synthesized by a former postdoc based on polyacrylamides. The idea is very similar. We have triboxspermine, and then we also have decylamine. So this is a saturated and not a non-saturated side chain. And also this postdoc performed a lot of characterization in terms of encapsulation, dynamic light scattering, zeta potential, to understand what formulation, what ratios do we need for efficient encapsulation. And here we saw that in the air-liquid interface model, we were able to outperform all commercially available transfection reagents. So interestingly, these polyacrylamides very well encapsulate and then also deliver the nanoparticles through the mucus, so that now we have an 80% gene silencing in this really relevant and almost pessimistic model of kalu cells that produce a lot of mucus. And with this, we were very optimistic and moved on into animal studies and also patient-derived tissue experiments. After we saw that in the animal experiment, most of the particles were taken up by epithelial lung cells, we looked into targets and decided to collaborate with a group in Gießen that is specialized into IPF, lung fibrosis. And from this group, we get patient tissue from fibrotic lungs. So these are lungs that usually transplant patients, and very fibrotic tissue, very difficult to deal with tissue. And in these fibrotic tissues, we were able to not only deliver the RNA, but really cause a very strong downregulation of a target gene called PAR2, and downregulated the gene to a much stronger extent than with any commercially available transfection reagent. But not only this, we were able to see that after downregulation of PAR2, we saw a very significant downregulation in collagen production. And this is really a therapeutic goal that we have for patients suffering from IPF. So as a little treat, at the end of my presentation, I want to show you this image. So this is fibrotic lung tissue. The fibrotic tissue has strong autofluorescence, shown here in yellow. You see the cell nuclei stained with DAPI in blue, and then the RNA in red. And I hope you can appreciate that with our approach, we can deliver the RNA into almost every cell within these precision-cut lung slices, which certainly is one of the reasons why we see such a good therapeutic effect ex vivo at this point. So with this, I come to the summary and end of my talk. There's still a lot of work for us to be done. I mentioned that we have a few AI approaches that are really helping us in developing new materials. We're working with a lot of ex vivo and 3R models, but of course also with in vivo models to mimic biodistribution and study things we cannot do ex vivo, really with the aim of developing nanoparticle-based nucleic acid therapeutics. And with that, I'd like to thank my group. I'd like to thank the very generous funding from the ERC, but also federal funding and many other funding agencies that are supporting all of these projects, and my team, the people that do all the work, and I'm happy to take any questions.
Brilliant. Thank you very much, Olivia. So the floor is open for questions. I saw Mark over there first.
Hi. Okay. Really nice presentation. My question is about your nano and micro particle approach. Are you using any approach to control the release of the nanoparticle from the microparticle?
So we really use this embedding into an amorphous or a crystalline matrix. And so the challenge actually is the colloidal stability of the nanoparticles. So I sometimes jokingly say it took us 3 PhD students to finally get to the point that we were able to optimize this, where we can redisperse the matrix into a suspension that has the same characteristics compared to the freshly prepared suspension. And there is no, let's say, controlled release mechanism in a way that this would be erosion-based or this would be degradation-based. But we definitely see that due to the fact that we have a dry powder that needs to take up the liquid, the lung lining fluids within the liquid, within the lungs, and we don't have a lot of liquids, or we shouldn't have a lot of liquids in the lungs, it takes some time. So we definitely have a delayed release. Sometimes when we add the dry powders onto the air-liquid interface for cell culture experiments, we actually see that it takes some time for, particularly for the crystalline solution, crystalline matrices to solubilize. And with that, we definitely see a release delay, but it's not really controlled by any other way other than dissolution of the solid matrix.
Okay, thank you.
I saw a question. Yeah, the person in the green.
Okay, thank you for your talk. And just following the previous question, I was thinking if you observe any change in the stability of your system just because of the confinement effect within the microparticles, and if, according to you, this could be a feasible approach to study and exploiting confinement effects on different kind of system. Thank you.
So we've done cryo-TEM imaging of the nanoparticles before and after spray drying. We currently don't see any differences. As I mentioned, we initially dealt and struggled a lot with aggregation and not being able to redisperse the nanoparticles into a good colloidal suspension. After optimizing spray drying parameters, process optimization, and so on, we're now able to redisperse the particles into the same hydrodynamic diameters. However, we see that there sometimes is what we call a lactose or sugar corona. So after the redispersion of the solid dosage form, we sometimes see that sugar molecules are still sticking to the surface. And we've done stability studies over 18 months. We see that the bioactivity and colloidal stability can be maintained for up to 18 months. But we do see that we sometimes have a bit of a sugar corona on the surface of the nanoparticles.
Okay. We have a question from Beppe.
Hey, Olivia, here. You left.
Sorry.
Very nice talk. Thank you very much. I want to pick up on your rationale for improving endosomal escape. And particularly, specifically to Sirna, right? Because you're probably familiar with the work from the Anderson-Langer group, where they did this really, really large screening of many, many different lipid nanoparticle polymers, etc. And they did it both for Sirna and messenger RNA. And it turns out that the two formulations are very different. And the formulation that work for Sirna, they tend to be very bad at endosomal escape, or much worse than the formulation that works for mRNA, right? We have similar data in our group. Generally, when we optimize endosomal escape, it works very well for DNA and mRNA, but Sirna has always been a puzzle. So what is your take of it? So do you think you really need endosomal escape, and perhaps it's not endosomal, maybe it's lysosome, it's a different stage? What do your data tell you about that?
So we've seen exactly the same, and it has puzzled us for more than 10 years now, I guess. And I mean, initially, I came from a plasmid transfection and then compared plasmid DNA with short interfering RNA. And I think it really has to do with the stiffness of the short double strand. So you don't have the flexibility that you have with a messenger RNA or even a plasmid. So I think the encapsulation is different. We're now essentially looking into this with simulation and comparing really the encapsulation, because we really think that it has to do with the particle formation initially. But it definitely also could be a matter of endosomal escape. That, I mean, the problem with messenger RNA is that if it's cut somewhere, then it's not effective anymore. With a short interfering RNA, first of all, it's more stable, stabilized by the double strand, so it's not so susceptible. So if it stays longer in the endosome, or it even moves into the lysosome, it's protected better, in my opinion. Whereas if messenger RNA ends up in the lysosome, I would assume that it's simply degraded and it's not active anymore. So I think a short interfering RNA complex, or a particle that remains in the endolysosome for a longer time, is probably, or might still be effective, whereas a particle that contains messenger RNA that doesn't escape from the endosome as soon as possible then is degraded and loses efficacy. So I think it really has to do with the topology of the RNA, and that then causes differences in encapsulation, and then probably also in intracellular processing.
Okay, we have time for one short question.
Yeah, so it was an amazing presentation. In fact, we have developed a similar technology, what we call nano-embedded API into microparticles of excipient for the same application. Then what we actually use spray drying, what you see is that you've tried to scale, then your particle size will actually go up, and then you may actually damage by temperature. So we have developed a process with this technology called high throughput electrospraying, where you actually process the whole thing at room temperature, and then you can stay in the 1 or 2 micron range. So we're happy to collaborate on that if you want, but my question was, so you said that when you actually process the API into these microparticles, that you have no issues with the stability, etc. So you actually found that you could survive using this spray drying process, right?
Yeah, so it was really, as I said, 3 PhD students optimized these process parameters during the spray drying process. And we have different sizes spray dryers, so we can scale up to a certain extent. We're also talking with a few companies that would be able to help us scale it up even further and see, obviously, if we run into the same issues, yes or no, or how we can, again, adjust this by a smart process optimization. But yeah, with the current processes, we can avoid colloidal stability problems. So we can spray dry into dry powder, and then when we redisperse, so if we dissolve the matrix excipient, we will get the nanoparticle suspension that is comparable to or not significantly different from the nanoparticle suspension that we have before spray drying.
Okay. Thank you very much, Olivia. And
thank you.
Join me in thanking her again.